I will finish this and then we just go for a quick break because the last section is just very, very important. Okay, so we're going to. This is just a refresher. Okay, since we've been through our lecture yesterday, right? Brain cells. Okay, the first thing, of course, is our neuron, right? Okay, yeah. So I talk about these things. So we had about 100 billion neurons. Okay, so name the different components. Okay. Okay, the first is a cell body, just a refresher, right? I'm sure everybody knows this. Then dendrites. Okay, I want to talk a little bit about dendrites later. Okay, then axon and our axon terminals, right? Which is I think is not too clear in this diagram. Okay, in our textbook, I think it should be collaterals. For me, I will put it as collaterals or axonic branches. I would say, yeah, but. The terminals usually is this little endings here, right? Yeah. Terminal, I mean, this are like the bulbs here, right? So actually the arrow is not that precise. Okay. So it's a fault of the publisher. Okay, it's not really anybody's fault, right? So dendrites here, right? So I mean, I would actually refer to it as a dendritic branch, right? So usually people like to just refer to it in a very casual fashion. Anything that kind of protrudes out of the soma, out of the cell body, and receives, nervous signal, they call it a dendrite. But then again, our dendrite, it's like the bowl of a tree. Remember that? And then we have so many small little branches that branches out. Okay, so and those things, we call them dendritic spines, right? Okay, spines. Okay, and then how do you kind of, balance all this terminology? So should you call them dendrites as well? Okay, or should you just stick to this terminology? You're pointing to this main branch of it. So for me, my preference. I mean, there's no consensus in the scientific community. I mean, unfortunately, because dendrites are super small, and then you really need to do very, very fine grained staining, right? So yeah, silver staining, unfortunately, Golgi staining, yeah. So usually I would, to be really precise, if I were to test anybody on this, okay, this thing would be the dendritic branch, right? So and then all the other one would be the so-called dendritic spines, and collectively they will be called dendrites. Okay, so take it as something like as a common component of this entire thing, this entire branch with all these mini branches. You call it dendrites. Yeah. Okay. Okay, so which cells in the nervous system support the neurons? Right, yesterday, okay, so this is glia cells, okay, or microglia, okay. Don't pronounce it as glia, right? Some people pronounce it as glia, okay. No, sorry. That's a person's name, yeah, okay. So it's glia, okay. So glia cells, okay, yes, refresher again, right? So we have our Schwann cells, okay, in our PNS, right, okay. And then oligodendrites are in our CNS, okay, in our brain, okay. Always remember that, right? So astrocytes, yeah. So I spent a lot of time talking about it yesterday. Very, very important, okay. You can't get this thing wrong. I mean, if you're showing a diagram, nobody will get the astrocytes wrong. But maybe it's just the oligodendrites and Schwann cells. People have some difficulty distinguishing between them, right? So microglia, I'm not going to talk too much about it. Okay, just take it that it's a cleanup crew. It's something to get rid of all the unwanted substances, blood. Okay, get rid of the neurotoxins. Okay, important. Yeah. The bulk of the research has been on the first three cells. I mean, astrocytes is just attracted so much attention over the past few decades. Right. Yeah. So again, this is the quiz, right? Okay. So the first, I show everybody the answer, okay, which cells provide myelination in the central nervous system, oligodendrites. And then this means that our Schwann cells, right, has to be involved, right, in our PNS, okay. Again, this is just a refresher, okay. Okay, so neural communication. So here I will just take a quick break before we carry on. So it's a very important part, okay, of extension of what we covered yesterday, right? So I will just stop for a while, and then I will give everybody a few minutes, okay? 3 minutes, okay? 3 to 4 minutes for a toilet break, and then we will continue, all right? All right, welcome back, everyone, okay? Yeah. Okay, just be a bit more quiet because I'm going to play the video, right? I hopefully that will not affect the quality of the recording, okay? So I'm going to stop the recording for a little while. Alright, so this first video, right? Okay. Right, this first video here, right, just gives you a very generic, overview about nervous transmission, right? So I just want to talk a little bit about what was mentioned, like the strengthening of the signals across the synapses. So if we get like repetitive, transmission of neurotransmitters, right? So that there will be a strengthening of such synapses, right? So in more scientific terms, we call it like a kind of Hebbian coupling, right? So it's just named after Donald Hebb, right? So that's really an integral foundational concept of neuroplasticity. So that's something to read about. Okay. I'm not going to elaborate too much on that until we get to the learning and memory lectures, right? So neural communication, right? So this is just a refresher course, okay, about what we covered yesterday, right? So we talk about action potential here, right? So which we are seeing on this slide, okay. So in a nutshell, right, it's just a mechanism for neuron to communicate with one another, right? That is what we call the action potential, right? So at rest, okay, all this thing is, revision from yesterday, right? Inside of the cell membrane is more negatively charged than outside of the cell, okay? We always remember, inside is always more negative. Then, we get this so-called negative 70 MV, right? And resting membrane potential, okay? Again, this is just emphasis of the same information, okay? Inside of the neuron is 70 millivolt less than the outside, okay? So we're talking about differences between two potential differences, okay? So it's differences between two pieces of different values. Always take that in mind, okay? They're not really absolute values, they're actually difference values, right? And then it's maintained by sodium-potassium pumps, okay? So some person asked me about how this pump, is it like one pump in charge of pumping sodium out, and then another pump in charge of pumping sodium in, all right? Unfortunately, right, for most of our textbook, this information is not clear, okay? It is not clear. But to my knowledge, okay, unless I'm hugely mistaken, right, it is usually one pump, okay? One pump should be able to, should be able to be responsible, right, for such movements, bidirectional movement of these two kinds of ions. Yeah. Okay, so but not to get too fussy, too meticulous over such nitty-gritty information. All right, so you just need to know, right, about that such pump exists, right, sodium potassium pump exists, and then we need it, for maintaining the resting potential, right, at negative 70 millivolts. So yeah, again, right, so the next slide, neurocommunication, right, it's a revision, all right, so information received by dendrites, okay, sodium channels open, always is a sodium channels, right, don't think of any other thing, right, so sodium ion channels flow into the cell. And then I think the second part, right, is what a lot of people will kind of forget, okay, despite I say it like so many times, it's like we need to reach this thing at a certain threshold, right, a certain threshold has to be reached, okay. So initially what happens is that your sodium ion channels will open, and then, a certain number, a quantity of sodium ions will enter, and then that raises your negative 70 millivolt to negative 65, negative 60, right? Depends on the strength of the stimulus. And then once that is reached, okay, that is the threshold, that is like the so-called invisible threshold, that has to be reached. And then if that is not reached, okay, that means you don't really have a lot of sodium ions, sodium ion channels opening. Okay, you will not have an action potential. So it's only when you have to surpass that threshold, negative 65, negative 60, then you will get this action potential, right? So that is what we call the all or none phenomenon, right? It passes the threshold, activation threshold, right? We have the action potential, right? If it goes slower, right? No matter what happens, there's no action potential, right? Keep that in mind, right? So yeah, so during depolarization, okay, the reverse happens, okay, the inside becomes more positive, okay, relative to the outside, okay, it's just direct opposite of what we have during the resting state. Yeah, so then the sub-bullet point depolarization of voltage-gated channels, right, means that sodium ions will flow into the cell, right, okay, and then this will affect all the adjacent sodium channels, okay, and that will move the wave, the signal, depolarization across the membrane, and we get the full-fledged action potential, right? So yesterday I think I did mention, but I think I rushed through it a little bit, right? So the action potential itself, right, usually it lasts between 1.5 to 2 milliseconds, okay, take note, okay? And after that, right, the refractory period is about a further 1 milliseconds, right, before we go back to the resting state, right? Yeah. So just take note of that, okay? Okay, so how to restore resting membrane potential? Okay, we need to restore the internal negativity of the neuron, okay? So it's a kind of repolarization, so we're going back to the baseline, okay? So depolarize, you're becoming more positive overall, and then repolarization, you're becoming back to the baseline, which is negative, right? So we have to get potassium out of the cell, okay? And then the question here is, right, which image shows depolarization, repolarization, resting membrane potential, right? Yeah. Okay. Oh wait. Before that, right? So we will, we will answer this question, right? Okay. Yeah. Let me just go back, right? So, okay. So, so depolarization is okay. I mean, it's spiking up. Okay. So repolarization is coming down. And then the last thing is, of course, the resting potential is always like a line here. So, not the greatest of all questions, but I just want to point out, right? So, this panel here, right? The right figure, sorry. So the first part, of course, that's a resting state. I mean, the flat line, and then we go up. Okay, that is actually when we actually have, you know, the middle box here. Okay, so that's like the middle part of the depolarization phase. That is actually when we have more sodium ions opening. It is don't confuse it. If I want you guys to label, I don't confuse it with say, okay, that is when the first few sodium channels open. It is not, okay? So we have a lot more, right? Because we already have the action potential started, okay? So it's, you know, they're already open, they're highly activated. And this part, okay, the bottom part, right? Okay, the bottom of this panel, okay, is that when you actually the inactivation, I mean the closure of this sodium channels have already happened, okay, and this is when you actually have the opening of your potassium ion gates. They're already open, and your ions are basically rushing out of the cell, right? Yeah, to restore the intracellular negativity. So this video here on action potential will show you everything, right? So let me just stop for a while. So everybody, just welcome back. So so for this action potential video, right? So what you get to see, right, is that there's this anomaly. I'm not so sure whether everybody saw it, but the resting potential is at negative 60 millivolt, right? Yeah. Okay. So yeah. So that's a little bit of an anomaly, and why is that, right? Because like yesterday, what I said, like negative 70 millivolt, it's a kind of statistical average. Okay. It's not taken as granted. I mean, not all our cells, not all our body, neurons, have that resting membrane potential, right? So, but I think what usually we kind of refer to, usually like the activation threshold, right, is always like 10 millivolt more positive, 5 to 10. Okay, that is kind of understood, yeah. So again, why exactly is like that? Okay, we have no idea. Okay, we are just scientists, biologists. They just observe the phenomenon, they record this thing, do it repetitively, and then accept it as the truth, right? Okay. So later on, I will go through this thing in slightly more detail. Okay, not to worry. Okay. So yeah, so diffusion, right? Electrostatic pressure. Yesterday I talked a little bit about it. Okay, they happen side by side. Maybe I didn't really emphasize this thing enough. Okay, so so if you really want to describe it, generation of action potential. If I say what are the two forces, you have to talk about the two forces that's involved. Please mention both. Okay. Okay, so permeability. Okay, just to remind everybody, this membrane, right, neuronal membrane is semi-permeable. Okay, so it's a plasma lipid layer. Okay, it's not totally transparent. Okay, and as a result, we have all these chemical synapses, chemical gates, right? Yeah. And so this video about forces to move ions in and outside a cell, okay, can be a bit complicated. Okay, I will need to explain this thing a little bit. Okay. All right, so everyone, so for this video, right, this forces to move ion in and outside a cell, okay, I really want everybody to just take note, right, of the importance of electrostatic pressure, diffusion, the two forces, okay, that's one. And then the other thing, just focus on the movement, all right, focus on the movement of the sodium and the potassium ions. So the chloride ion bit, you know, is a bit confusing, okay. It is a supplementary component of action potential, but it's not like the primary, you know, driving force. Like diffusion of chloride ions, it's not a primary driving force. So if you're being asked a question about this, about action potential generation, you know, forces involved, okay, I will not force everybody to write about chloride ions, okay. That's a bit too much, okay. But personally, I would just say that just take note, right. So initially in the extracellular environment, we do have more chloride anions, okay, negatively charged anions, and they do move into the cell, okay, they do move into the cell, okay, yeah. All right, so notes of Ranvier, okay. So the next slide, let's just carry on, okay. Notes of Ranvier. Yesterday I talked about it, okay. So notes of Ranvier are important for saltatory conduction. Remember, okay, leaping conduction. So that will really accelerate the speed of signaling, right? Nervous signaling. And everything will start at the axon hillock, okay? Don't get it wrong, okay? And our myelin sheath, right, is made up of Schwann cells, okay? Yeah. All right, so again, a refresher, okay, electrical synapse versus the chemical synapse. So electrical synapse, remember, is less common, much less common than the chemical synapse found in our heart, okay, found in our organs, okay, that really requires immediate action, okay, heart, lungs, right. Not so sure about our lower torso. I think it's mainly chemical synapse, right, if I'm not mistaken, right. So, and then also pay attention to the size of the gap, okay. The gap itself is 3.5 nanometers. Okay, remember this, okay. Very small gap, right. And then in your next video, you will see that they're pretty much attached to each other. And then you can hardly decipher, you know, which is the dendrite and which is the synapse. Okay, yeah. So that's where the point between where is the dendrite and where is the axon. Yeah, that gets a bit murky. Yeah. So chemical synapse, just a quick refresher. Okay, in order for it to work, before we actually had the release of neurotransmitters. So yesterday I talked a bit about calcium channels. So we actually need to get calcium in. Okay. There's calcium in the extracellular fluid, okay, along with sodium cations. We need to get them in, right, so that they can, you know, get involved in all these chemical reactions. That's kind of driven by this organelle we call the Golgi apparatus. So Golgi apparatus will actually manufacture the vesicle itself. So the vesicle are all the spherical blobs, you know, with cerebrospinal fluid again, okay, and then it will cover all this neurotransmitters, okay, neurotransmitters that reaches the presynaptic terminal, okay, and then after that they will just release it, or you will fuse it with the presynaptic membrane, release it into the synapse, and then that's what's happening, right? And that really contributes. It's the neurotransmitter that's really flowing across the synapse, okay, that region, that really produces that initiates, right? I wouldn't say that it's ultimately the creator, but it actually initiates, right, the action potential, right? Okay, so, and then we also have some receptors. I'm not so sure. Maybe I talk a little bit about it, right? We have something called an autoreceptor that's actually involved in the reuptake, okay, of all these neurotransmitters, okay? And we'll take a look, okay? We'll take a look at this last video, and I will elaborate more, right? All right, so welcome back, right? So this last video of ours is just, I think, in my own, you know, personal opinion, I think it's fantastic, right? Yeah, actually it's very accurate in depicting this electrical synapse, right? That's what it actually should look like, right? Yeah, you can hardly see the synapse itself. Yeah, it's pretty much glued together, right? The two terminals, right? Presynaptic and postsynaptic terminals. So that's something to take note of. And also, for some of you, if you're like kind of interested in drugs, you know, for your paper, right? This video series is something you can refer to, right? Okay, very, very useful. Right, so the last component, right, of today's tutorial, I just talk about in detail, right, about what the synapse is about. Okay, so again, refresher course, right? So it's action potential, right, transmitted to adjacent postsynaptic neuron and the synapse, right? Essentially, it's just a point of connection, right? Sometimes people they call it synaptic cleft, right? As what the video just now told us. Okay, that is more precise actually, right? But then again, this is very general synapse. Okay, we just talk about any kind of connection. Irrespective of the space. Okay, as long as there's this point of contact. Okay, between cells. Okay, we just refer to it as a synapse. Okay, so electrical and chemical synapse. Right, the two broad categories. Right, and electrical synapse very, very fast again. Right, so and its gap is it's very, very small. Okay, 3.5 nanometers. Okay, extremely fast signal. And it's found in our heart, found in our liver. Right. Okay, and surprisingly, it's found in plants. Okay. Yeah. I take that as true. Okay, I take that as true from the textbook. Yeah. And chemical synapse. Okay, this is what we have been focusing on, right, for the vast majority of this entire lecture and tutorial series. Right. So. It's really driven, okay, modulated or mediated, depending on how you look at it, by our neurotransmitters, okay. And neurotransmitters are, of course, released from the presynaptic cell, okay, sending cell, okay. And then the sending cell, specifically what it is the axon, right? Yeah, it is the axon, right? So, and then neurotransmitters, okay, bind to the postsynaptic receptor cells, the receiving cell, right? It should be the dendrite, right? The dendrite of the receiving cell, okay. And then the chemical signal is then terminated. Okay, once all the neurotransmitters got to the right places, and of course it's slower than electrical synapse now, right? Remember, because it's chemically driven, right? And then we have two kinds of signals. Right, one is excitatory, one is inhibitory. Okay, and then this kind of signals allows for selective activation, variety of responses. Okay. Depending on the specific location on action of the receptor for different chemicals. Okay. So again, we have this release of neurotransmitter diagram. Okay, this is just slightly enlarged version. Right. So synthesis of neurotransmitter. I talk about it. Right. That's driven by our calcium ions, and then storage in vesicles. All right. Axon terminal, and then exocytosis. You just need to remember the word exocytosis. Okay. It's just we are releasing it, okay, and then this phase, the subsequent phase, is action on receptors, okay. So they will diffuse, they will bind, right, with the dendrites of the receiving cell, the receptors, and then the deactivation. Okay, that's a bit more interesting. That's just like our internal, way of recycling all this biochemicals. So our body actually has this amazing way of recycling them, and then in a way it actually conserves energy, right? So this is actually pretty important. So if the autoreceptors, somehow couldn't really re-uptake, what happens, for people who are on drugs, the autoreceptors get dysfunctional, right? So that is why they will have all this, behavioral abnormalities, right? So if you're on cocaine, that really affects, okay? Basically you have an overdose of certain neurotransmitters in your synapses. And so two broad categories of our receptors. So one is ionotropic and metabotropic receptors, okay? And both kinds of receptors are chemical receptors, okay? They're not electrical, okay? Even though we see the word ion somewhere, okay? But don't get it wrong, okay? So because the ion is, the ion channels are letting the ions in, right? Okay, so two types of receptors. The first one, right, so it's ionotropic receptor. So tropic means like, you're kind of attracted to something, right? So the neurotransmitter binds with an ion channel. And then this is just a one-way, a one-pass mechanism. So once it binds to it, right, the ion channel opens, right? And the other ions, will kind of flow in, right? So this is a one-way, okay? Right, and then the second part would be, so what exactly those transmitters are, right? We'll talk about it in our next lecture and tutorial, okay? That would be much more fine grained. So metabotropic, okay, receptors, okay, meta means, it's related to metabolism, okay, this is a two-way procedure. So the first thing the neurotransmitter comes in, okay, it does not, after you bind it with the receptor, it will not open this ion channel immediately. Okay, what happens is that it will send out activate the synthesis of the G protein, which is a kind of, intracellular messenger, right? It works a little bit like our calcium ions, right? And then that would kind of, propagate another kind of signal telling the ion channels to open. And then, that's what happened at the second stage. And then that thing will open, the ion channel will open. All right, so that's, you have an additional intermediary stage. Yeah. So again, right, so which type of synapse make use of ionotropic or metabotropic receptors? What I said just now, right? All right, chemical, right? Yeah, don't get this thing wrong, okay? Yeah. In a more complicated version of this question that you see, okay? Don't get it wrong, yeah. Okay, so this is important, right? So what happens when neurochemical binds to postsynaptic receptor, postsynaptic potentials, okay? So two outcomes, right? One is excitation and one is inhibition. So excitation, you just, increasing the likelihood, okay, that the postsynaptic cell will generate an action potential, all right, okay? And inhibition, you will just reduce the likelihood. Simple as that, all right? Think in terms of probabilistic terms, all right? Statistically speaking, okay? It's not all or none, okay? Don't think in terms of all or none, all right? So, and then we have two kinds, right? One is EPSPs, okay? And then IPSPs, okay? So these two kinds of information is highly important, right? If you somehow cannot make it for the midterm, have to sit for a deferred paper, right? I know a few people will have to do that. So this, this component will be important, right? Yeah, because it will be a different paper and some questions will be different, yeah? Okay, all right, so, but anyway, you got to know this, right? So this slide input is combined equals to neural integration. What is it showing, okay? So I think it's a few pages, right, in our in our textbook, right, chapter 3. It's not that easy to understand, actually. It took me a while, just to be honest, and I really have to go back and revise a lot of things, okay? So, all right, so let me just show everybody the bullet points. So we have 4 here, right? So the first one everybody understands, right? We receive inputs from many neurons, right? So from the dendrites, the cell body, right? So how this thing, right, the EPSP or IPSP is generated is that the cell body itself, right, or maybe like a main dendritic branch, right, it receives this axonic terminal, right, that very, very close to its to the cell body or the dendrite, and then that thing actually fires. Okay, that thing is receiving the signals that's being propagated by that particular terminal. And then within the cell itself, right, and then you have a lot of chemical, biochemical reactions happening within the receiving cell body, and that really kind of propagates the signal all the way to the axon hillock, right? And that is where you will get, the regeneration of the action potential. So that's how it actually works. So it's not like something that you shouldn't be taken as something like a firing of the action potential. Action potential is just within the cell body itself, right? It has to receive something. The cell body itself has to receive something from elsewhere, right? So always keep that in mind. And so the first panel, right, the left panel, right, so the EPSP alone, okay, it happens slightly before the action potential. All right, take note of that. It's very hard to spot, okay? It's a slight depolarization, right, slightly before we get this full-blown activation of the action potential, right? So this is what we see here is, both right, EPSP and the action potential starts at -70, right? And then this one is kind of, EPSP. Once you have the AP started, right, once it crosses the threshold here, which is -65, then, right, after that, then you have the EPSP spiking up, right? It has to pass the threshold, so it actually kind of, it's there like spiking already, but then the spiking of the EPSP itself, right, it happens together, with the activation threshold, right? Yeah, so, and then it spikes upwards, usually for, about 10 to 15 millivolt, positive millivolt, yeah? And then after that, it goes down, right? Like a sharp gradient, it just goes down. So that's the EPSP itself, right? So IPSP is like the reverse direction. So instead of going positive, we go negative, by about 5 millivolt, okay? So it's just a slight, polarization. You don't use the D, a slight polarization. So going negative. And then in the event of IPSP, always remember, once you have IPSP, there cannot be an action potential, right? There cannot be an action potential, right? So this is a part that a lot of people will not kind of follow, yeah. So I just want to mention this, okay? So IPSP alone is just IPSP, right? No AP. And then when this EPSP and IPSP, when they're kind of so-called integrated, wow! I mean, our neuron can actually do math. So this is the amazing part, right? So ultimately they will summate back to negative 70 millivolt or somewhere around there, right? It really depends on the scale of depolarization and polarization, but it will sum to the baseline, right? So that's an amazing feature, yeah. Over a span about 2 to 3 milliseconds. So an amazing feature that our neuron can do, yeah. Okay, so the last portion bit, right? So I talk about this thing, right? Neural integration, but it's also subdivided into two kinds, right? One is spatial summation, one is temporal summation. So do take note of it, okay? Spatial summation, very easy to understand, right? So we have many, okay, presynaptic terminals, more than one, okay? And then basically it's affecting the receiving cell body, right? And then that will start off a new action potential, right, at the axon heel lock. Okay, so basically multiple presynaptic terminals, okay, they call it spatial summation. So if we have just one presynaptic terminal, okay, then that thing fires repeatedly over a length of time, okay, that's temporal, time related, then that's what we call temporal summation. And temporal summation is actually important for the formation of those what we call Hebbian synapses. Synapses that are really strong. That really channels nervous signal that that's kind of derived right from certain parts of the brain. Yeah, so that really strengthens the connectivity between certain synapses. Yeah, so this is a special feature of temporal summation. Yeah. All right, so I think this should be the last slide, okay? And then we just talk about neuromodulation. So this is a key word. So when we talk about one neuron affecting another neuron, so this is just neuron to neuron interaction, okay? We talk about neuromodulation. It's really at the neuronal level. It shouldn't be taken as between different brain regions. I know it has been misused, but the right use of this word is neuron to neuron interaction. And then usually, we have this exonic synapse, okay? Because we do have two, at least in this diagram, right, two axons interacting with each other. So just need to take note of the first thing called facilitation, right? Presynaptic facilitation, and then another one we call it presynaptic inhibition, right? So so that's really before, right? We actually all these neurotransmitters get transferred across the synapse. So yeah. Actually, it's kind of hard to see that because we do have a few synapses here, right? We have one first synapse here, right, before neuron, between this neuron C and neuron A, right? That's the first synaptic cleft, right? So where is actually the presynapse, right? I mean, it's kind of some kind of a misnomer, right? Yeah, but take it as it is, right? So so by this pre, right, we're actually referring to all the axons before, the secondary synapse, okay, the second one here. Okay, the second one that's right in front of neuron B, right? So basically, you have all this, neurotransmitter going on, right, between C and A, and then that really kind of facilitates some kind of signal propagating through this neuron A, and that kind of tells neuron A to send out more, okay, not less, okay, more neurotransmitters to pass through the synaptic cleft, leading all the way to neuron B. Okay, simple as that. That's just facilitation. Right, you're facilitating neuron C and neuron A, just facilitating output of more neurotransmitters. And then for presynaptic inhibition, we're not doing it. Yeah? We have a neuron D this time, okay? The neuron D is the inhibitor, right? So it's sending out all these inhibiting neurotransmitters. So yeah, we'll cover them in our next lecture. So what are all these transmitters? Inhibitory neurotransmitters. And that will tell neuron A to kind of kind of produce less of a certain kind of neurotransmitters, which can be excitatory, which can be inhibitory, yeah, either way, right? So as a result, neuron B doesn't receive a lot of inhibitors from neuron A. So that is inhibition. Yeah, very simple as that, right? All right, so test your learning, right? So the last slide, okay. I wouldn't really go through all the questions one by one, but I just want everybody to focus on the last 3 questions, right? They are a little bit tricky, okay? Focus on the last 3 questions. They will be very helpful, okay? Very helpful for your preparation for the midterm exam, okay? All right, so that would be all for tutorial 1, okay? So for those of you who have questions, yeah, look through your notes and come and ask me, right? Okay.